working on the transcriptional regulation of IL-2 in macrophages. Then he moved to the East Coast and did a postdoc with Dan Littman in NYU. There, he discovered, actually made a few really important discovery, including identifying ROR gamma T as a regulator for Th17 and demonstrating that Th17 represents a novel T cell subset that are critical for a variety of human diseases. In 2009, Liang started his own lab in Northwestern in Chicago and was promoted to tenured associate professor in 2015. He was then recruited to the University of Florida and served as a professor and as an associate chair for research in the Department of Infectious Diseases and Immunology, and also the director for the Center of Inflammation and Mucosal Immunology. So Liang's lab has made multiple influential discoveries in characterizing the molecular interactions among transcription factors that govern the differentiation and developmental programs of T cells and ILCs. For example, he showed the regulation of ILC3 and ILC2 by transcription factor AHR, STAT3, and ICAROS. He also revealed a dual function of Helicobacter in regulating ILC number and function. On the other hand, interestingly, he also found ISO3 can promote salmonella infection. So in today's talk, I think Liang is going to tell us some recent work going on in his lab on the metabolic and the nutritional regulation of gut immunity. Without further ado, Liang, the floor is yours.
Okay, you guys can hear me, right?
Yes, yes.
So thank you so much, Shelley, for the kind introduction. And actually, you already gave the talk, so I don't need to talk about too much. So really, I want to use this opportunity to describe our efforts to understand mucosal immune responses, especially during the past couple of years work on a new area for us, especially the metabolic and the nutritional impact on those immune cells in the mucosal tissues. And so I want to quote a very cliched saying called "food is medicine." And I'm really a true believer that what you eat, you are what you eat. Basically, the diet and the nutrition have a great impact on our cells and our immune system as we are immunologists. So our lab has been really fortunate to study this problem or answer this question from a mechanistic angle by focusing on environmental sensor called aryl hydrocarbon receptor. I'm going to talk about it in details today during the 15-minute talk. So you probably all know this, the ILCs. My lab has really intrigued by ILC biology. Especially, we are interested in type of ILCs called group 3 innate lymphocyte cells. And the reason is, as Shelley mentioned, that I'm interested in ROR gamma T, and ROR gamma T is expressed as a marker for those group 3 innate lymphocyte cells. And those cells are very important, play very important roles. Some of them are really important for lymphoid organogenesis. But in adult, those cells are important for antibacterial infection, for example, and also involved in recently shown by Greg Stoltenberg and others that have anti-tumor immunity. And then those cells, if not very regulated well, can cause inflammation, can cause autoimmunity as well. And then ILC2s, those cells are secreting IL-13, IL-4, and IL-5. and important for allergic reactions. But recently also there are data showing that at least in humans, ILC2 can have anti-tumor immunity as well by directly lyse or kill tumor cells. And as I mentioned before, my lab really interested in this molecule called hydrocarbon receptor. It is a ligand-dependent transcription factor. In the absence of ligand, AHR is tapped in the cytosol. Once binding to the ligand, AHR translocates into the nucleus and heterodimerize with its partner called ARMS or HIF1 beta. Then this dimerization, the protein complex will bind to the AHR responsive or dioxin responsive elements that induce array of genes, including prototypical AHR target gene, CYP1A1 or AHRR, and also immune genes such as IL-22, cell cycle genes, P27, and survival genes, and BAX, etc. And there are two types of ligands for AHR. One category ligands called xenobiotic ligands. Those are environmental toxin and those things you can find in the air pollution and or in various toxins can activate AHR, such as dioxin. But endogenously, AHR is a molecule really very well conserved. So it should not just detect a toxin, right? So yes, it has physiological function, and there are endogenous ligands that have been found to activate AHR potently at the similar level of similar potency as dioxin or TCDD. Among them, FIXI is one of the most famous ligands for AHR, which is a tryptophan derivative. And also there are other ligands derived from phytochemicals generated by digesting vegetables, or microbiome such as lactobacillus can metabolize tryptophan to generate AHR ligand. So therefore, we think that AHR is really a key mediator of this dialogue between environment and host, and is a fascinating molecule to study because its expression and activity can be regulated and therefore have a different impact on the immune responses. So when I started my lab, there are two really seminal papers published by Gita Stockinger's group and Howard Weiner's group that showing that AHR actually play a role in Th17 cells and also Treg cells. But those experiments are mainly using in vitro culture and in vitro cells. So when I entered this, established my own lab, we asked the question is, how can we study this in vivo or in a cell type specific manner? So there are challenges and approaches. So typically in the field, there are pharmacological approach. Basically, you can use mouse diets to either eliminate AHR ligand or adding back AHR ligands such as phytochemicals and other tryptophan metabolites. Then you can add into the diet to feed the animal to activate AHR. Or you can use AHR knockout, straight knockout, but better using AHR conditional knockout that you can delete AHR in a tissue-specific manner. So all these things is to really ask the question about the function of AHR in the cell type specific manner. But the challenge is that when you're using this activation approach by pharmacological administration of chemicals or diets, etc., you will activate AHR in many cell types. So to bypass or to circumvent this problem, Zhou, a former postdoc in the lab, actually developed a knocking mice that is called, we call it AHR care mice. Basically, we knocked in AHR constitutive active form of AHR into the AHR endogenous locus. And this preceded by flux stop cassette. So in the absence of a Cree, this is a knockout because there's no transcription. And But when we cross these mice to a cell type or tissue specific cree, then we can delete. This stop cassette allow expression of constitutive active AHR independent of ligand, and also mark those cells by GIP. So we can not only mark those cells by GIP, but also activate the AHR specifically in the cell type and tissue specific manner. So using those tools, we over the years have really tried to answer the question of what is the AHR in the mucosal tissue in the gut. So there are lots of work in the field, but I would just use it because of time. I'm just using this slide to summarize over 10 years work from just our own lab, try to take a peek at the real AHR biology in the gut. So Zhou showed that AHR actually play a very important role in the ILC3 maintenance. And she showed that AHR gamma T interact to promote IL-22 expression. And together with Xiaohuan Guo in Yangxin Fu's lab at University of Chicago at that time, we showed that STAT3 together with AHR can promote IL-22. So all this is very important for the control of microflora. The IL-22 can stimulate epithelial cells to secrete antimicrobial peptides, and those without ILC3s, without IL-22s, you see outgrowth of those, pathogenic bacteria. But also you can see outgrowth of commensal bacteria called segmented filamentous bacteria, SFB. And as a result, the Th17 cells in the AHR knockout is upregulated. Th17, you can see more of Th17 in the AHR knockout animals. And later, Shiyang Li and Jennifer Heller in the lab showed that Icros can suppress AHR activity by disrupting AHR-armed interaction. Therefore, Icros actually function as a suppressor in ILC3. And in adult mice, when you knock down Icros, you can see increase of ILC3s. And later, Shiyang further showed that AHR plays a cell-intrinsic role in the ILC2 to suppress ILC2. Therefore, we think that AHR actually also as a modulator to modulate the ILC2 and ILC3 balance in the gut. And John Bostic, a grad student, showed that Helicobacter, certain Helicobacter species can have a dichotomous role on ILC3. On one hand, it activate ILC3 to produce IL-22, but on the other hand, it's also inhibit ILC3 proliferation and decrease ILC3 numbers in the gut. So recently, Zhou Fu, a postdoc in the lab, showed that IL-22 can actually direct inhibit type of epithelial cell, tough cell differentiation in the gut. And those cells are now gaining a lot of interest because they can produce IL-25, etc. So that's just a 30,000 feet overview about our study, and that sets the stage for our future research on the role of AHR regulating in the immune responses. So today I would like to share with you three stories. One is dealing with a cell-intrinsic role of AHR in CD8 T cells, which is not very well studied. And then talk about the role of R gamma T positive gamma T17 cell and ILC3 in the tissue homeostasis and infection. So Joseph, a graduate student, he graduated a couple of years ago. He was really interested in the idea about whether AHR play a role in tumor immunity, because he's coming from a tumor immunology lab, and CD8, especially interested in whether AHR play a role in CD8 cells, because AHR has been implicated in tumor immunity, and there are a lot of complex data out there some data suggests that AHR in the tumor cells is a tumor promoter. When you activating AHR, you will promote tumor. But there are also data suggesting maybe AHR could be anti-tumor. So we like to take the advantage of, I mentioned before, AHR flux mice to look at this more carefully. So we breed this to CD8 crete, and you can see here that when knocked out AHR in CD8 cells, the tumor using B16 tumor model, the tumor grow much bigger. And in those mice, we saw that triple positive CD8 cells, polyfunctional CD8 cells are reduced. And this probably contribute to the impaired anti-tumor immunity in those mice. And using single cell, you can see that, you can appreciate that indeed that some population, for example, those polyfunctional 4 population, 5 population, and 6 are all reduced in AHR knockouts if we establish a B16 over model, B16 melanoma model. And but then there are cells, actually we saw the circulating memory cells actually upregulated in those tumor infiltrating cells. So it's suggesting that AHR is important for tumor resident CD8 cells and the TRMs, and especially for those TRM function. And to look at the gut, as I mentioned, that we are also, where main focus of the lab is really in the intestine. So we look at the gut. We also observe that in the gut, in the small intestinal IEL, we saw a decrease of CD69, CD103 double positive cells, TRM cells, and perform a gene set enrichment analysis. Zhou further showed that the knockout has decreased resident core gene signature, but gained the circulating core gene signature. As you can see here, then knockout, the cells are, TRM genes are really reduced, but then TCM genes are upregulated. And by integrating of ChIP-seq data using in vitro TRM, AHR ChIP-seq, and the RNA-seq data using IEL resident CD8 alpha beta cells, we looked at the AHR directly regulate genes. As you can see here, that AHR directly activate those tissue resident genes and then repress those TCM genes and also the circulating core gene, the signatures. And just to summarize this, because it's published, we show that AHR activation can promote TRMs to really directly, we have evidence show that AHR to promote those gene transcription, and then AHR suppress other gene transcription. In the tumor environment, AHR really promoting a triple positive granzyme B, TNF, and interferon gamma positive polyfunctional CD8 cells, and promoting anti-tumor immunity. So just switch gear a bit to talk about the ILCs and our gamma T positive lymphocytes. So Zhou Fu from a postdoc in the lab really interested in the gamma T cells, and as you can see here in the mouse, there are at least 2 types of gamma T cells based on the CD44 and CD45 staining, and also interferon gamma IL-17 expression. Those are gamma T17 cells. They express IL-17 and they express our gamma T, and also gamma T1 cells express interferon gamma and express T-bag. So these cells are for example here in the spleen in the mouse gut, and also you can find those cells. And metabolically, the gamma T1 and gamma T17 cells are different. So deep red staining to measure the mitochondrial mass and potential, you can appreciate that gamma T17 cells have much higher mitochondrial mass. And also TMRE, the mitochondrial membrane potential, is much higher in gamma T17 cells compared to gamma T1 cells. And the cellular loss level is also higher in gamma T17 cells. So this suggesting that there are metabolic differences between gamma T1 and gamma T17 cells. And we looked at this further by looking at a key transcription factor for mitochondrial biogenesis called TFAM. And you can see that gamma T17 cells have higher TFAM mRNA expression, and accordingly have more mitochondrial DNA content and more mitochondrial DNA gene transcription. And on the other hand, glucose intake measured by 2-MBDG and glycolytic gene expression measured by those genes are not much different between gamma T17 and gamma T1 cells. So this suggests that there are metabolic differences. And with this, we looked at ATP production, and actually gamma T17 cells make more ATP than gamma T1 cells. So TFAM, as mentioned, is a key transcription factor for mitochondrial biogenesis. It's important for mitochondrial DNA packaging, transcription, and replication. So to ask the question about mitochondrial function or mitochondrial metabolism in the RGMT positive gamma T17 cells, we, again, using genetic tools, basically we cross TFAM flux mice to TCR delta Cre ERT2 mice, and we can invisibly delete TFAM in gamma T cells. So as you can see here, that the R gamma T positive gamma T cells, gamma T17 cells, are drastically reduced. However, the gamma T negative cells are not reduced at all when we delete the TFAM using TCR delta Cre. So this suggests that TFAM is important for gamma T17 cell maintenance or generation, and consistent with the idea that gamma T17 cell have more mitochondria respiration, like TFAM concentration, mitochondrial gene transcription, etc. So to carefully look at the R gamma T positive cells, the TFAM role or mitochondrial role. So we cross this to the R gamma T Cre to delete TFAM in R gamma T positive cells, including gamma T17 cells, ILC3 cells, Th17 cells. And you can see here that the mice, when we delete TFAM, on week 1, there's no difference in gamma T17 and ILC3s, indicating that the TFAM or mitochondria is not important for the gamma T17 or ILC3 generation. But when the mice aging, like mice are week 3 age or week 6 age, we saw a drastic reduction of gamma T17 cells, as you can see here. And ILC3 cells also reduced at week 6. However, in sharp contrast to those cells on the week 6 and later on, Th17 cells, no difference between the control and the knockout, suggesting that really, although they are all R gamma T positive, but they really have different metabolic dependence on mitochondria, for example. And those knockouts, the gamma T17 cells not only decrease in the gut, small intestine, but also decrease in all different other organs. You can appreciate, the spleen, lymph node, liver, lung, fat, skin, etc. But there's one organ actually at a time we could not get. That's small intestine, because previously I showed you this is large intestine data, but small intestine we cannot get it. So Zhou really puzzled and struggled with that small intestine gut product, but later he found out that actually the mice, the knockout mice, have much longer small intestine, and this lengthening of small intestine actually correlates very well with the age of the mice. When mice aging, the small intestine get longer and longer, and the histology showed that actually those mice have more inflammation in the small intestine. However, in the large intestine, no inflammation, nothing, everything's all good. So that's probably the reason why he cannot doing small intestine gut prep to get in lymphocytes to examine the gamma T17 cells easily. So because the mice getting older, they will develop inflammation and get longer intestine. So we are really intrigued by this tissue remodeling because this intestinal length typically is pretty resistant to modulate to getting longer and shorter, etc. So there are two types of immune cells I mentioned before are decreased in the knockouts, right? So in the T5 knockout, those are gamma T17 cells and ILC3 cells. So to ask the question, which cell type contribute to this phenotype? So Zhou did the bone marrow transfer experiment. And you can see here that when we transfer the bone marrow marked by CD45.1, we can restore the ILC3s here, ILC3s very well, but we cannot restore gamma T17 cells. So as quantified here, the bone marrow can restore at least very, a big extent the ILC3 cell, but the gamma T17 cells cannot be restored at all, still decrease drastically in the T5 knockouts. And now we ask a question about the intestinal length. As you can see here that those mice with the bone marrow transfer with restoration of ILC3s, but not gamma T17 cells, still have longer intestine similar to untransferred mice. So quantify here. So suggest that ILC3 by itself is not probably not sufficient to restore the intestinal length. So recently, Zhou further did experiment using TCR delta mice knockout PFM and showed that they mimic the R-gamma mice have longer intestine. So this is really give a hint that maybe gamma T17 cells are really responsible for this phenotype. So recently, Zhou, a grad student, and Eric Helm, a postdoc in the lab, did the definitive experiment by transfer the bone marrow and fetal thymocytes together because it has been shown that the fetal thymocytes can generate gamma T17 cells in the gut. And by doing this co-transfer, actually, we can bring this intestinal length back to normal. And you can also appreciate that when we transfer this fetal thymocytes, we can restore the R-gamma T positive gamma T17 cells to the wild type level, even better than wild. So with this, we think that indeed the gamma T17 cells are very important, and probably together with ILC3 are required for restore the intestinal length. So several years ago, Rich Loxley's lab at UCSF published a paper showing that the dietary fiber can promote a pathosymbiont trichomonas and to secrete succinate. Then succinate can promote tough cell and activate them to secrete IL-25. Then IL-25 can enhance ILC2s. So then there's a tough cell ILC2 circuit. As a result, producing large amounts of IL-13. Then IL-13 can promote the goblet cell type cell hyperplasia. And as a consequence of this hyperplasia, they show that the intestinal length is become longer, intestinal lengthening, a very similar phenotype to our mice. So we therefore asked the question about what about our mice? Do we see this kind of circuit or not? So we did an RNA-seq of a small intestinal tissue, and indeed we saw the tough cell gene is upregulated, and also goblet cell gene is upregulated in the knockout mice. And this is quantified by qPCR, and also this is by qPCR, and also by confocal look at protein expression of BCLK1, which is a marker for tough cell. And so therefore indicating that those mice have more tough cells, and the more tough cells may be responsible for this small intestine lengthening. And looking at the tissue, we also observed the elevated type 2 immune responses, both ILC2s and Th2s. And those Th2 and ILC2 type 2 immune responses, we think, are responsible for tissue lengthening, tissue remodeling, and the small intestine lengthening, because the data suggests using anti-IL-4 or especially anti-IL-13, we can shorten the intestinal length to the wild type level, as you can see here. So this is indicating that indeed the type 2 immune responses are very important for this tissue remodeling and for this lengthening phenotype observed in the TFM knockouts. Then those mice, not only have small intestine lengthening, but also they have better anti-helminth immunity, as you can see here, that using H. pylori infection model, we show that the worm burden and the eggs are all decreased in the knockouts. Mechanistically, how this works? So we think at least one mechanism is through IL-22. Through collaboration with Christian Jobin's lab at UF, we show that using an organoid culture system, IL-13 can induce BCLK1 expression. You can see here, can induce BCLK1 expression on tough cell differentiation. But when we adding IL-22, we can really inhibit this BCLK expression. And IL-17A has no impact. And this is a FACS data. You can see that IL-17 does not inhibit BCLK1 expression, but IL-22 completely shut down the BCLK1 expression. So this suggests that IL-22 suppress BCLK1 expression, suppress tough cell differentiation. Then without IL-22 in the knockout, because IL-22 is gone due to lack of gamma-17 cells and ILC3s, we see a hyperplasia of proliferation of tough cells. And indeed, the IL-22 knockout has also increased small intestine length and increased tough cell genes, etc., and type 2 immune responses as well. And to ask the question whether indeed the IL-22 is responsible for this phenotype, so we performed IL-22 plasma hydrodynamic injection, basically put back IL-22 into the knockouts, and we can show that we can decrease type 2 immune responses, but also decrease the tough cell differentiation or tough cell gene expression, and the small intestine length, it becomes shorter. So indeed, the IL-22 we think is one of the key elements in this story, in this regulation. So to summarize this, we show that hopefully you can appreciate that in the wild type mice, the gamma-17 and ILC3 can secrete large amount of IL-22. They can directly inhibit tough cell differentiation and suppress this tough cell ILC2 circuit. But in the absence of TFM or in the absence of those cells, the IL-22 decreased. Therefore, you have a enhanced tough cell ILC2 circuit, and then caused the small intestine tissue remodeling and lengthening. So finally, I want to use last about 20 minutes to talk about a recent story really performed by postdoc Li Fengxiong in the lab, and he actually was interested in the iron effect on the gut immunity, and especially ILC3. So basically, he fed the mice, the wild-type mice, with either controlled diet or iron deficient diet. Then looking at the lineage negative R can be positive cells, ILC3s. He showed that in the small intestine, he saw a decrease both in the small and large intestine, and the cells produce less IL-22 and IL-17, as especially IL-22, you can quantify it here. So this really suggests that iron maybe regulate ILC3 generation and also ILC3 function. So he looked at this more carefully by ex vivo approach. Basically, he isolated small intestinal ILC3s, then treat those cells with iron chelator DFO, and in a dose-dependent manner, you can see that DFO decreased the IL-22 expression drastically, as quantified here. Because ILC3s are very challenging to do biochemistry, to ask doing metabolism studies, etc. So we sort an approach to really try to study this stuff in vitro. So MNK3 cells is a pretty good cell line that is a model system for us to use to ask a question about ILC3 biology, because they produce IL-22. As you can see here, they produce large amounts of IL-22 in vitro. So similar to ILC3s, primary ILC3, adding DFO greatly reduced IL-22 expression and also IL-17 expression. So again, suggesting that iron is important for ILC3 function. And to carefully look at the gene expression on a global scale, and at a global scale, we perform an RNA-seq, and to compare plus DFO and minus DFO MNK3 cell gene expression. And you can see here that there are quite a genes are upregulated when adding DFO, including the GLUT1 gene involved in glycolysis, glucose intake, and HIF1 alpha, etc. But also a lot of genes are downregulated. Those genes are, you can see here is IL-22 I mentioned before, the AHR expression, IL-17A, R-gamma T, KIT. So those are all ILC3 signature genes. So by looking at the pathway analysis, we also identified that the DFO greatly reduced the oxidative phosphorylation genes. As consistent with this, the mitochondrial genes are all greatly reduced when adding DFO in the MNK3 cells. And using seahorse, we looked at the oxidase OCAR and the ECAR, the oxidase and glycolysis. And you can see here that when adding DFO, the MNK3 cells, the OCAR, representing oxidative phosphorylation, are decreased, and however, the ECAR are increased. So this data suggests that the iron depletion impair mitochondrial function by decreasing all the mitochondrial genes, etc., and switch the metabolism from oxidase to glycolysis in MNK3 cells. And to ask the question about how iron impacted ILC3s, so we asked whether the iron transporter play a role. If we delete the iron transporter, what will happen? Can deleting iron transporter mimic iron deficiency or DFO treatment? As you can see here that indeed, when we knocked out TFRC, which is CD71, which is a transferring receptor involved in importing iron into the cell, when we delete the CD71, we saw a great reduction of ILC3s in both small and large intestine, as you can see here. And this reduction is also, we think, is a maintenance, because at week 1, no difference, but at week 4, we start to see a great reduced ILC3s, both in the large and small intestine. And this is due to a proliferation defect. As you can see here, the knockout mice ILC3 has a drastic reduction of KIC7 staining and also BRDU incorporation, suggesting that the proliferation of ILC3s is impaired upon ablation of CD71. I want to point out that different from ILC3s, steady-state Th17 cells are not affected by knocking out CD71, as you can see here. No difference at all between wild type and knockout. However, during Citrobacter infection, we saw that the Th17 cells are greatly reduced. And this, we think, suggesting at least 2 implications. One is the metabolic status or metabolic requirement between steady-state Th17 versus the infectious Th17 cells are different. Second is, during infection, the Citrobacter will compete with the host and will consume large amounts of iron. And therefore, maybe Th17 cells in a very limiting iron condition, the TFRC or CD71 actually play a very important role. So that is, need to keep in mind that in the iron sufficient condition, Th17 cells probably not, important. But if you eliminate iron to a certain level, maybe the Th17 cells require CD71 for its proliferation or for its maintenance. As a result, those mice, the knockout mice, have reduced ILC3s, as I showed before. But when we feed the mice with iron to doing iron rescue, we can really rescue the ILC3 number. You can see here, those ILC3s are restored greatly in the, especially both in the small and large intestine. You can see here. So this suggests that the iron indeed is the key element for ILC3 maintenance, and if you overload the mice with iron, you can rescue it. And then the mice without iron, the mice die, body weight drop, while dying from Citrobacter infection. But if the knockout mice giving iron, they can be rescued. No dying, no cell death, no weight loss. And the bacteria count also show that without iron, you have much higher bacterial load in the feces. But when you put iron back in, you can reduce this bacterial load. So this really prompted us to ask the question, the mechanistic question about iron supplements. What's the gene expression changes? So you probably can appreciate that this is the knockout mice without iron treatment. This is the wild type mice also without iron treatment. This is basically steady state, normal diet. They're very far from each other. The gene expression totally changed. And one of them is the ILC3 signature genes. So the TFRC genes have reduced ILC3 signature genes. But when the mice fed with iron, you can see here, now the mice are all clustered together, knockout and wild type. So suggesting that the iron can rescue majority of gene expression defects. And one of them are ILC3 signature genes, as you can see here, the false detection rate Q value, significance value is 0.25, and here is 0.376. It's not considered significant. So it's basically restored all the defect of ILC3s, consistent with the idea that your ILC3 numbers can restore in the intestine. And to carefully look at the gene expression, we noticed that there are around like 1,800 genes between conditional knockout and wild type. They are differentially regulated genes. And those genes, majority, when you feed iron, you decrease it, you disappear. So those 1,600 genes, you can see that before iron, they are different, right? But once you feed the iron, the wild type knockout, no difference at all. And among those genes are such as like glucosa genes, HIF1 alpha genes, etc. But more interestingly is this 479 genes that appeared in the differential genes upon iron administration. And you can see here, there are 479 genes, and among them, for example, those genes are upregulated in the conditional knockout. And those genes are representing, for example, DNA replication cell cycle pathway, consistent with the idea that those cells, the knockout cells, why they can restore, in the knockout mice, the cell number can restore is due to the increased DNA replication and increased cell cycle, consistent with the higher proliferative activities of those cells. And this also suggests, this iron overloading experiment suggests that there are other transporters than CD71 involved in incorporating or transporting this iron into the cells. Among them, ZIP14 is one of them. We think is a non-transferring binding iron transporter. As you can see here, we generated TFRC as ZIP14 double knockouts, and to knock out CD71 and ZIP14 together, we can decrease the ILC3s significantly, even more than the single knockouts. And then more importantly, the ILC3s, you can see here the iron can rescue the CD71 single knockout, but cannot rescue the double knockout. So suggests that indeed there are other non-transferring binding iron transporters, and one of them is ZIP14 play a very important role. In the absence of CD71, ZIP14 may be responsible for this transport of iron into the cell. And I want to come back to AHR regulation because as I mentioned before, the lab is really interested, and during our interest in AHR and during our ChIP-seq and also RNA-seq analysis, we saw CD71 is a target gene. And indeed, when we knocked out AHR in RGamma positive cells and in ILC3s, we see an upregulation of CD71. And when we activate AHR using AHR care mice, we see a downregulation of CD71. So suggesting that AHR cell intrinsically inhibits CD71 transcription in ILC3s. And indeed, this is also, you can see here from using string knockout, the AHR knocked out in all cells, you can see that the decrease of ILC3s and then the iron, when we put iron in, we can actually enhance ILC3. So the idea here is that when you knock out AHR, CD71 is upregulated, allow those cells compensatorily to incorporate more iron into the cell, and then promoting the cell proliferation, and then promote more ILC3s. So indeed, you can see here that the ILC3s in the AHR knockout, when we put iron in, we can really increase the ILC3s in those mice, and increase the Ki67 proliferation in those mice as well. And mechanistically, you can see here there is a single peak of AHR binding, this by AHR ChIP-seq at the TFRC or CD71 locus promoter specifically. And when we knocked out the AHR, and you can see that the transcription, the RNA-seq transcription is going way higher, much higher compared to the wild type, consistent with upregulation of CD71, and consistent with the idea about AHR as a suppressor of CD71. Then I want to point out that using ATAC-seq to see the chromatin structure or chromatin remodeling at the promoter, especially, we can see that there's no difference between AHR wild type and AHR knockout, suggests the AHR is not involved in chromatin remodeling at this locus. To further look at this transcription regulation, we did the AHR overexpression in the MNK3 cells. We showed that the AHR wild type can suppress CD71, but AHR mutant that cannot bind DNA cannot suppress CD71. So all this together indicating that the AHR is a suppressor of CD71. And I feel obligated to talk about the microbiome, and because as a mucosal immunologist, if you don't talk microbiome, will not be appropriate. So we actually Li-Feng find an interesting phenomenon that in a germ-free mice, we see actually upregulation of CD71, especially in the large intestine, consistent with the idea there are more bacteria in the large intestine. And however, no such thing in the Th17 cells. So the germ-free mice, Th17 cells, CD71, no change at all. And this upregulation of CD71 can also be recapitulated by antibiotics treatment. You can see that the CD71 is upregulated here, is upregulated when wild type mice treated with antibiotics. You can see here, right? Then AHR knockout have increased CD71. And this increase not further upregulated by antibiotic treatment. So we think that the microbiome inhibits CD71 expression by ILC3 specifically in an AHR-dependent manner. So right now we are trying to figure out what kind of bacteria involved in this process and what bacteria generate what ligand. We hypothesize that the bacteria probably generate ligand to activate the AHR. So I want to put this all together in a model slide that we hopefully with this to convince you that the iron is a very key nutrient element for ILC3 maintenance, especially for ILC3 proliferation by modulating ILC3 mitochondrial function and the metabolism. And this is really involved in the AHR regulated feedback loop. Basically, AHR suppress TFRC expression, but in the AHR deficient background or in AHR deficient animals, TFRC is upregulated, allowing to incorporate more iron into the cell, therefore to rescue at least partially ILC3 defect in the AHR knockouts. So with that, I would just stop here, and I think I mentioned all of those people's names, and then also my collaborators, and finally my funding situation. So once again, thank you very much, and thank you everybody. Stick around till almost 9 p.m. and Eastern time, and I will take questions.